In this podcast, we're going to look at Helicobacter pylori. Particularly, we're going to look at how Helicobacter pylori can survive and proliferate in the stomach and cause peptic ulceration. Before looking at Helicobacter pylori in more detail, I just want to pause for a moment and highlight the fact that there are many bacteria present in the human body. So particularly in the lower intestinal gastrointestinal tract, there are many bacteria present. Over 100 trillion bacteria is the estimated number, which means that there are about 10 times more bacterial cells living in the human body than there are actual cells in the human body. Quite an amazing number. In the upper gastrointestinal tract, there are fewer bacteria present, although there's quite an abundance of bacteria in the mouth. For instance, every milliliter of saliva contains millions of bacteria. But by contrast, the esophagus and the stomach contain far fewer bacteria, probably because these areas are washed out very frequently. There are many types of bacteria that can cause stomach upsets. So for instance, here on the left are just a few examples of bacteria that can give rise to nausea, vomiting and diarrhea, in addition to other symptoms. However, these bacteria here on the left are only able to survive for a short while in the stomach and are unable to colonize the stomach long term. By contrast, Helicobacter pylori has the ability to survive for a long term in the stomach, and this bacterium is able to colonize the stomach as well. And these characteristics, the ability to survive long term in the stomach and the ability to colonize the stomach, are really unique characteristics of Helicobacter pylori. Helicobacter infections are very common, and usually individuals are infected during childhood, for instance, when they eat food containing Helicobacter pylori. But although they are infected at an early age, symptoms do not normally appear until adulthood. However, not everyone with Helicobacter pylori infections develop symptoms, and these so-called asymptomatic infections are not well understood. It may be that these infections are asymptomatic because of characteristics of individuals, for instance, because of their specific characteristics of their immune system, or maybe because of the types of sugar chains present on the gastric epithelium. It could be because of a specific strain of Helicobacter pylori is less likely to give rise to symptoms because not every Helicobacter strain is identical, and there could well be other unknown factors that we are as yet unaware of. So what does Helicobacter pylori requires in order to survive and colonize the stomach? Essentially, there are four things that they require. First of all, an acid neutralizing system, which allows them to deal with the hostile environment of the stomach. Key enzymes here are the urease enzyme and transporter proteins that we'll look at in more detail, particularly because this system is exploited to diagnose Helicobacter pylori infections. So in addition, they need adhesion molecules so that they are not washed out the stomach. And these are present on the outer membrane of the bacterium, and they recognize, for instance, sugar chains present on the gastric epithelium. They need movement. Helicobacter pylori are equipped with flagella, which allow them to move to the epithelium surface. And finally, they need to degrade the mucus that promotes also the movement to the epithelial layer, and this is provided by lipases and proteases that are secreted by the bacterium and that cause damage to the mucus. And this is, of course, resulting in the damage of the protective lining, causing eventually ulceration. Let's now look in more detail at the acid neutralizing system of Helicobacter pylori, which is so important for diagnostic tests. So this is a schematic diagram of Helicobacter pylori. Helicobacter is a gram-negative bacterium, which means it has two membranes, an outer membrane and an inner membrane. Now the space between the outer and inner membrane is called the periplasm, and it is normally the pH of this periplasm is normally around 6.1, and the acid neutralizing system has two proteins that play a key role. One of them is the urease enzyme, which is a very abundant enzyme. Up to 10% of all proteins in Helicobacter pylori could be a urease molecule. And the second protein is this so-called proton-gated urea channel, which is localized on the inner membrane of Helicobacter pylori. And so together these two proteins form an essential component of this acid neutralizing system. So this slide shows how these two proteins work together to neutralize the acidic conditions, or to survive in these acidic conditions. So this system requires urea, which is normally present in the stomach, and the urea can enter the periplasm via poorly characterized mechanisms. Once it is in the periplasm, it can be transported into the cytosol across the inner membrane by the action or via the proton-gated urea channel. Once the urea is in the cytosol, it will be converted into ammonium and carbon dioxide by the action of the urease enzyme. Both ammonia and carbon dioxide are gases. They can diffuse back into the periplasm across the membrane by simple diffusion. Carbon dioxide can diffuse back to the outside of the bacterium, where it can cause this bloated feeling and belching. Part of the CO2 can enter the circulation and be exhaled. And the ammonia is a base and can neutralize protons from the gastric environment and neutralize the pH in the periplasm by the formation of ammonium. And so these two proteins, the proton-gated channel and the urease protein, play a key role in this neutralization process. The urease enzyme is quite an amazing enzyme. It's very large. It contains two polypeptide chains, the alpha chain, which has a structural role, and the beta chain, which contains the catalytic center. So these two chains in total form this symmetrical ball-shaped enzyme that contains in total 24 polypeptide chains. So 12 of these chains are alpha chains, 12 of these are the beta chains that contain the catalytic site. The enzyme is very large. It is over 1 million Daltons in size, in weight, which is about half the size, or just under half the size of a bacterial ribosome, but for instance bigger than the RNA polymerase, which make the RNA from DNA. So it's an amazingly large enzyme for a relatively simple job. The beta chain contains the catalytic site, and it requires a cofactor. It contains 2 nickel ions for every beta chain, and so the beta chain is an example of a metalloenzyme. It requires a metal as a cofactor. The proton-gated urea channel contains a single amino acid chain um, that forms a homohexameric ring-like structure. So here you've got these six um, chains of the proton-gated urea channel that are identical and form this ring-like structure. So this is a side view, so you can see it's a sort of a flat disc-like shape. Now the interesting thing is that these channels, which are actually located here, are fully open when the pH is very acidic, 5 or lower, but they're closed when the pH is neutral or alkaline. And so in this way, the proton-gated urea channel prevents the periplasm to become too alkaline. And this hole here in the middle is actually filled with lipids. So this is just a closed area. So the urease enzyme and the proton-gated urea channel work together. And their action is really specific to Helicobacter pylori. And therefore, the detection of this enzyme system is the basis for diagnosing Helicobacter infections. The principle of the test is to provide the individual with urea that is labeled with a stable isotope that is naturally occurring at a very low frequency. So this allows the urea that is given to the patient to distinguish it from normal urea present. And by the action of these two proteins that I've just explained, the proton-gated channel and the urease channel, the urea is converted into ammonia and carbon dioxide. And this carbon dioxide, at least a fraction of it, can enter the circulation and eventually can be exhaled by the individual. So a sample is taken just before urea is taken and after 30 minutes or so. And then the presence of this labeled CO2, which is derived from the ammonia that was given to the individual, is detected, can be detected by a variety of techniques. So all the types of tests, and you might still see some literature about this, contain this carbon-14 label, which is a slightly radioactive isotope, which can be detected by scintillation counting. And the most commonly used test now is to use the carbon-13 label, which is non-radioactive and can be detected by either mass spectrometry or by a special type of infrared spectroscopy, which is in fact more convenient and quicker to get results. So understanding the acid neutralizing system should help you to understand the principle of the urea breath test. And it should also help you to understand why it is recommended that an individual should not take any antacids just before the urea breath test. It should also help you to understand why the urea that is presented to an individual is in a solution of citric acid that is very acidic. So in both cases, of course, it is related to the action of the proton-gated urea channel. Now, the urea breath test is the most commonly used test to diagnose Helicobacter pylori. It is, of course, of interest to note that there are other types of tests available as well that are very specific and sensitive. Those include the blood test and the stool tests, which are using these very easy to use strip tests. And apart from these non-invasive tests, there are invasive tests which involve gastroscopy and the taking of a biopsy for culture. So that's shown in this picture here. So this system, this test, the so-called CLO test, also is based on the presence of this acid neutralizing system of Helicobacter pylori. So in this case, Helicobacter will be incubated here in this medium containing urea. So the medium is very acidic at the start, and it contains a pH indicator. So if Helicobacter pylori is present, the medium will be neutralized, which can be detected by the presence of the pH indicator.